There are going to be some things in Chapter 2 I want to talk about, but not a whole lot. Let's see. Color vision, we talked, I think neodynia is where we left off, if I'm not mistaken.
Yeah.
So basically neodynia is your brain keeps growing after you're born, so that's like super exciting, right? So I'm glad everybody has that to look forward to. Here is just evolution of the primate species. We already talked about the difference between apes and monkeys, right? And that's the tail, so great job on that. Here's comparisons of brains. Can't really see what's going on here, but this is humans. We have bigger brains. We talked about that. How many of you know about other species sort of in the lineage to get to Homo sapiens? We've got Homo erectus and Homo habilis. They have slightly smaller brains than humans, except like Neanderthals, right? So you guys know about Neanderthals, and that should be a tall, not a thal, right? So it's like the H is silent, just to throw that out there, because that's an important point. You don't want to sound like an idiot and call them Neanderthals when they're Neanderthals. Also not Cro-Magnon, right? Because it's a French word, right? So would you say Cro-Magnon? Yeah, there you go, right? So that's a lot better. And Neanderthal is actually German, right? So it's found in the Neander Valley, so that's why they get that name. So there you go. So those are slightly important points. Neanderthals actually had bigger brains than humans, which is kind of fascinating. We always like to, or than Homo sapiens, always like to think, well, like we have giant brains, so we're awesome. That's why we beat those guys out, right? In fact, there's some new theory that maybe they were like super nice. You know, they had bigger brains, they had bigger frontal cortexes, and so they were better about thinking of negative consequences and didn't want to deal with that guilt of like hurting other species, right? As much as maybe Homo sapiens does, because you know, we're actually pretty awesome at hurting each other. I don't know if you guys have noticed that, like at any time, right, Eric? I mean, we've got some interesting capacity for that, right? And if we came up against another species that was a little nicer, we'd probably win, right? So that's how that goes. All right, large skull, we've talked about birth canals. I should mention this briefly. Tons of stuff if we're going to do studies on humans or on animals. There are a lot of groups who are going to check on that. So all the stuff we're going to talk about for the most part is stuff that, I mean, obviously the stuff that happened in the 1800s, there were slightly fewer sort of oversight, going on, but now that things are in the modern times, anything we talk about, Emily's going to have like all these real checks and balances, right? Folks checking in on our studies to make sure they're up to date. Everything's going the way it should. All right, so no worries there. I do want to spend a little time in Chapter 2. Just a moment. Whatever. All right, now the computer's going to not let me click on buttons.
Did you enable editing?
I tried clicking on that, and it's not letting me click on that button either. The whole thing is just collapsed. Let's try that again. Brilliant. All right, so Chapter 2, what's cool about Chapter 2 is everything. So everything in here is going to be important, but we're not going to spend a lot of class time on it because we do need to move forward. This is related to communication within and between neurons. Lots of stuff here we don't need. Central nervous system, peripheral nervous system. There are differences. I do want to spend a moment here talking about these sort of 3 steps in your nervous system, right? I like to think of your nervous system as doing 3 things. It can handle input, it does some sort of calculation in between, and then there's an output. Okay? And that's really what's going on here with sensory, motor, and interneurons. They're not in the right order, right? And you can take this sort of 3-step process, the input, calculation, and output, and you can apply this to nearly every level of the nervous system, all the way down to an ion channel, which we'll talk about, and all the way up to an entire organism, right? So you can kind of zoom in and you can zoom out on this. Okay? For our purposes, we want to think about a sensory neuron, for example. That's clearly going to be input, right? You're gathering some information from the outside world, so you've got some input there, right? That's pretty cool. You've got to do some kind of calculation in between, right? Like, what is this input I'm receiving? How should I respond to that? You've got to think, you've got to plan movements. Right? There's a lot of things going on. You've got to access your memory stores. There's all of that calculation process happening. And then you need to respond to that, right? You need to make some kind of action, right, Jason? So you want to have a response. If someone throws a rock at your face, and you just make it to that calculation stage, it's not going to do you a lot of good, right? You're going to get hit in the face with a rock. Okay? So you've got the input, you're like, wow, there's like a giant gray thing flying at my face. So that's the calculation. You think, well, that's a rock. It's probably going to hurt if it hits me. Ray's over there calculating the momentum of the object based on the rotation, right? And you're just like doing all these calculations. But then you've got to make some movements. You've got to send a motor command like, I got to stick up my hand, I got to duck, I got to roll, run, I got to do something, right? So I have to dodge this. So that's what we talk about when we think about input, output, and then that calculation step in between. And we'll talk about calculations on a sort of smaller scale here in just a moment. Parts of a neuron, we've got a dendrite. Please don't call that a dendritic tree, right? That one kind of bothers me. It really does, and I know I mentioned this in other classes, but so dendrite is from the Latin word for tree, right? So if you call it a dendritic tree, it's kind of like calling it a tree, which I think is a little, is called dendrites. Dendritic arbor is slightly acceptable. So we have dendrites. You've got your cell body. That's the soma. Your axon, covered by the myelin sheath. This thing is going to increase velocity. And it's also going to increase fidelity, right? So it's going to prevent leak of that signal. So it's going to make sure that the signal that is sent is what is received from one neuron to another, right? So Travis, that's going to be important, right? If you're going to try to dodge that rock, you don't want halfway through sending this signal to your arms to like put up your hands to grab it for the signal to get lost and you just go, then you get slapped with the rock, okay? And that's bad. Down here we have terminal buttons. Sometimes you'll call those terminal boutons. People like to get really fancy with it. Terminals, whatever. They're all kind of interchangeable, so don't get too worked up about that. Here is sort of, that was a neuron in isolation. Here's a neuron not in isolation, right? So we have connections coming from other cells, going to other cells. This could be from the outside world. Or it could be some internal other nucleus sending information, right? Sending that information forward or so forth. We will talk a lot about neurotransmitters. We'll talk more about those when we get to chapter 4, so don't worry about that too much. Exoplasmic transport, we need to send things sometimes from the cell body down to the terminal. Sometimes we have to send things from the terminals back up to the cell body, right? And that's going to be anterograde or retrograde depending on which direction we're going, okay? We'll talk about stuff we'll send in a little bit. Hey, how many of you took biology when you were in high school and or here? Yeah, right, like everybody had to take a biology class, okay? You guys remember that cell, it had all the stuff in it like cytoplasm, had a nucleus, you've got your endoplasmic reticulum, right? You've got mitochondria. I was just waiting, nobody's going to say it, that's great. You've got the Golgi apparatus, you've got all kinds of stuff in there. Your neurons really have the same things, right? They have the same basic kind of parts. that any other cell would have. They're specialized, so they've actually chucked a lot of stuff out that we don't need anymore, right? And they've built up a lot of things that we do need. We're going to have a lot of mitochondria. Your brain uses a lot of energy, right? Okay. Probably like 25% of your blood flow at any given moment is going to your brain, which only weighs about, it only takes up about 2 to 2.5% of your body mass, right? That seems like a disproportionate amount of blood flow to your brain if it wasn't doing a lot, right? And it does a lot, again, it needs a lot of glucose. This is why every single one of you love Fun Dip, right? Everybody loves Fun Dip. There's nobody who's going to go like, nah, I don't want to eat Fun Dip, right? You might try to act like you don't want it, maybe you're on a really strict diet or something, but you're going to take it anyway, right? You're going to eat it in secret. And if it's not Fun Dip, it's something else, right? Some other kind of sugary substance. We love things that are sugary. Your brain runs exclusively on glucose, so it needs glucose to do things, right? To create all of that energy, we're going to feed that glucose into the mitochondria. We're going to get ATP out of that. I don't want to cause any flashbacks, so hang on. I'm going to say Krebs cycle. For some of you, that's going to be really frightening. For those of you who don't know what it is, just move on. It's like a ghost that lives under your bed. If you don't know it's there, it's not going to bother you. But for those of you that lived through the Krebs cycle, Eloise, you did that, right? No? Do you know the Krebs cycle?
Yep.
You do? Yeah, it was a nightmare, wasn't it? It's all right. It's not bad. The guy won a Nobel Prize for that. It's pretty cool, right? He won a Nobel Prize for torturing students for decades. So we're going to take that glucose, we're going to turn that into energy, and we're going to go. Hey, one thing that a neuron can't do, one thing it can't do is actually store glucose. Okay, so it's not going to have a place to store glucose inside of it. Your other cells can store up a little bit of glucose, right? And then we can use that when we need it. We're not going to be able to do that with a neuron. So where are we going to get our glucose? Later we're going to talk about support cells, glial cells, astrocytes in particular. How many of you know about DNA? All right, Ray, you were really excited about that. Appreciate your DNA enthusiasm there. Actually, only like 2 hands went up and yours went the fastest. So DNA, what we're going to end up doing is we're going to make proteins. This is important. What are proteins? So like basically anything in your body that does something, that's probably a protein, right? So we're going to talk later about ion channels. Guess what? They're made out of proteins. Made out of these things called amino acids. Okay. You've got a number of these amino acids, right? We've got to create that protein. It's going to be in some kind of shape based on the structure or the sequence of the amino acids. Some of them have positive charges, some of them have negative charges, some of them don't have any charge. Right, so they're going to maybe have them, maybe not. Based on those positive and negative charges, they're going to kind of roll up and clump up in a certain structure, and that means they can do something, right? Okay. I know that seems kind of broad right now, Eric, but just hang in there. It all makes sense later. So our ion channels, also enzymes. How many of you love enzymes? Yeah, everybody should, right? Without enzymes, guess what happens? Well, you die. So you got that to look forward to. But enzymes are going to be important. We're going to talk about neurotransmitter synthesis and breakdown later, so we're going to need enzymes for that. If these enzymes are screwed up, if these ion channels are screwed up, you're not going to be able to do the things that you need to do, okay? There are actually things called channelopathies. Okay. These are individuals whose ion channels are not structured properly. They don't allow for ion flow in and out of cells appropriately, and Travis, this can cause some problems with neural function, right? A lot of these channelopathies are actually fatal, especially when we're dealing with calcium. How many of you know a place other than the brain where you might have calcium channels? What, muscles? Yeah, and there's one muscle in particular, and it's getting to be that time of year. I'm just going to draw a little heart, because Valentine's Day is coming up in a few weeks, so you guys should be prepared for that. Your heart, right? So if you have problems with your calcium channels and your heart doesn't work, that's a bad thing. Oh, hey, here we go. Here are astrocytes. These guys are pretty cool. They can store glucose. They can put glucose in to neurons so the neurons can use those. So that's kind of awesome. They'll also help with some other processes. Oligodendrocytes are going to be awesome. We'll talk about those when we get. They create the myelin sheath. in neurons on the axon, so that was what, again, helps accelerate that electrical signal, right? It works as insulation. Microglia, they're actually responsible for the immune response in your brain. How many of you want to get a brain bacteria? No one. That's the answer. Nobody wants a brain parasite. Most of you might have one. That's kind of frightening. So you should think about that, right? How many of you have cats? Sorry. You probably have a brain parasite. But it happens. Don't worry about it. Yeah. No cats? Don't worry about it. Yeah. Your clothes are ironed too. That's like how you can tell on a guy. Like, so it's like a true story, right? So here's a little side story about brain parasites. So who wants a brain parasite story? In cat feces, there's a parasite, right? You guys are familiar with that, right? That's an L. Okay. What they'll do with this parasite is they'll try to actually infect rats with this parasite. So this is actually kind of fascinating. So they'll infect rats with a parasite, and that parasite will get in the rat brain, and it's sort of kind of weird. It sort of makes the rats like sexually attracted to cats. Which is, I don't know, I guess it makes the rat happy, right? It's like, okay, that's great. But what it really does is it makes the cat happy, right? Not because the cat's really like sexually interested in a rat, but a cat is metabolically interested in a rat, right? Because it can eat the rat. And it's much easier to eat an animal that's like trying to nuzzle up to you than one that's trying to run away from you, right? So that makes sense, right, Jason? So, like if I could make cheeseburgers sexually attracted to me, that'd be a lot easier, right? Because then they would just like flock to me and I would have to like go somewhere to get a cheeseburger. So that makes a lot of sense, right? So Matt's thinking about this. He's like, how can I get cheeseburgers? I really think I got hot stuff. Because I am just tired of walking down the fat patties. So think about that, right? And cats figured it out, right? So they put this this bacteria in their urine, or in their feces. And that's what it does to rats. It can actually infect humans as well. And it does. And in humans, when they get infected, there's actually some evidence that it makes them a little, like, sort of careless, right? And so they stop, like, paying attention to things. Like, they'll walk out into the middle of the street, like, more often than not, right? And they're actually involved in more car accidents. Like, people who are infected with toxoplasma are involved in more car accidents than people who are not, because they're a little more reckless, right? And then you would think, like, well, if I was a little reckless as a rat, then I might try to get it on with a cat, right? So that's pretty adventurous and reckless, right? If you're a rat, so that's people. At least in males who are infected with toxoplasma, they tend to dress very sloppily, right? So they won't like tuck in their shirts or iron them. They're all kind of things are crumpled all the time. So you can watch for that. So that's how you know, like if you're walking down the street and you see somebody and they're like kind of dressed sloppily, you might think, that guy's attracted to cats. I don't know, Eric. That's where my mind goes on these things. But that's relatively a true story. I may have stretched a little bit there on the, I don't think humans are actually like physically attracted to cats. I mean, at least because of this parasite. That's pretty exciting, though, right? Here you thought you were in control of everything, but this is cat feces. It's the small influences on your life that make a big difference, right, Jason? I mean, it's not like the big things. Like, you know the big things, right? But it's these small things that go unnoticed. And largely, we've not studied brain parasites. This is like a true story. We've largely not studied brain parasites, ever, and their effects on humans. We've studied them in other sort of situations, but we've not really studied like the way that brain parasites sort of on this low level might affect average changes in your behavior, right? Like just like in small. Like what if you were just slightly more likely to be careless, right? And think about like the changes in your behavior, right? If you're not like thinking through decisions clearly, right? I mean, just a small shift in, and we'll talk in a moment about membrane potential. Membrane potential could really cause a big difference in your brain function, right? So there you go. Matt's still back there writing down about cheeseburgers. That's a pretty good idea, right? All right. Here are astrocytes. Guess what? They're shaped like stars. That's cool. One of the reasons that you can't really get glucose in your brain without astrocytes is something called the Better Business Bureau. Also called the blood-brain barrier, right? It's 3 Bs. So the blood-brain barrier is awesome because it tries to keep bad things from getting in your brain and screwing it up. And you might think, well, geez, I know better than to do something like that. But you don't. Trust me, you don't. Because you do it all the time. And not only on top of that, not only do you not know not to put bad things in your body that could hurt your brain, you're smart enough to know what bad things to put in that actually will still get into your brain and make you do things that you probably shouldn't, right? So you've got your blood-brain barrier. What's awesome about that is it does prevent a lot of things from getting in your brain. What's not so awesome is anything that's lipid soluble will slide right past that guy, right? And what's like the #1 lipid soluble substance that people ingest?
Alcohol.
Yeah, alcohol, right? That guy goes right into your brain. And that blood-brain barrier it's not even like a turnstile. They just took it off and just open the gate, right? So you can just slide right through your cell membranes. So that's why alcohol gets in and makes you do stuff. It does awesome things, like it makes you more polite. It makes you more, it increases your athletic prowess, right? It makes you think your athletic prowess is increased, right? How many of you know someone, it's not you, know someone and when they've been drinking, they can instantly jump 10 feet farther than they've ever been able to jump in their lives, right? Because they're always, it's like, I can jump that. Like, well, you can't, but I'm willing to watch you fall in the hole. So there you go. I would recommend you stay away from alcohol. I think that'd be a waste of my breath. This is the point in the lecture where I try to give you that really good life advice. And that advice is going to be drink craft beer. Stick with me. It's more expensive, so you'll drink less of it, right? If you're buying Miller High Life, even a poor college student can afford enough to cause some damage. But if you're drinking some nice, small batch craft beer, most of you can't really afford enough of that to cause some long-lasting damage, right? So you're going to pace yourself. That's good advice, right? Abstinence doesn't work. So I don't tell people don't have sex, just tell them to wear condoms. It's a better approach. Craft beer, it's the condom of beer. It's a safe approach. Who's ready for that on YouTube? Jason's going to call his mom and go hey, guess what we talked about today? All right, oligodendrocytes. Again, we talked about that. They make this kind of jelly roll around the axon. Not a big deal. We talked blood-brain barrier. If you guys want more details on this, I would recommend listening to those lectures. We're going to skip over reflexes. All right, this is where we should spend a little bit of time, though, because I really want you to understand action potentials. Okay? And this is where it gets a little complicated. So we're going to spend a little bit of time talking about action potentials, and then we're going to breeze on to something else. So what is an action potential, right? It's this electrical potential. It's a change in sort of electrical charge across the membrane, right? And I know that might not make a lot of sense to you right now, but we're going to get there, so just kind of hang on, right? How we do this is we take what's called a microelectrode, which is going to be a really small piece of metal. And we're going to insert that into the neuron. Okay. And when we insert that into the neuron, we can measure what's the electrical charge inside the cell and what's the electrical charge outside the cell, right? And we can compare that. And that comparison is going to be our membrane potential, whatever that is. Just for arbitrary purposes, we're going to say that the outside of the cell is always equal to 0 millivolts. Okay? And whatever the inside is going to be plus or minus from 0 in comparison, right? It's going to be less than that or it's going to be more than that. The outside is always going to be 0. Now, what is outside of a cell? We want to think about that, right? Outside of a brain cell is mostly salt water. Okay? Lots of sodium, lots of chloride. Okay? Just a big vat of salt water. Okay? Got some cells in Inside the cells you have different ions. We'll talk about those in a moment. Okay, so outside it's going to be 0, inside it's going to be something else. If, and this is sort of a hypothetical state that let's imagine doesn't happen for an extended period of time. If that particular neuron, or whatever neuron you've decided to pick, 1 out of a trillion, you've got a trillion of them, so pick your favorite one. I know it's going to be a hard choice, right? You're going to offend almost a trillion other neurons, but pick one. If that neuron is not receiving any contacts, right? There's nothing telling that neuron yeah or no. Nothing's happening. It's going to be at what we call resting membrane potential. Now, this number is, depending on the book, I'm not exactly sure what it is in this book, it's going to probably be like negative 65 millivolts, might be like negative 70 millivolts, somewhere in that range, right? Okay, 70 is what's in the book. Some books will tell you 65, some will tell you 70. Doesn't really make a big difference, okay? It's in this ballpark, right around negative 70 millivolts, okay? That's going to be resting membrane potential. That's going to be that neuron just hanging out there, just waiting for some other neuron to tell it, hey, guess what? It's time for you to fire an action potential. Anytime we fire an action potential, anytime we do anything, we're going to try to get back to that resting membrane potential. Okay, we're going to work our way back to the resting membrane potential. That takes a lot of energy, and we'll talk about that in a moment. Here's some words you need to know. Depolarization. That's a movement towards 0. Okay, that's a reduction. We say reduction because we're moving towards 0. And you might think, if I'm normally moving towards 0, that's not good. But in this case, it is because we're making ourselves more positive, right? So anytime we make a cell more positive, that's going to be a depolarization. We're making it less polarized. Okay, we're making it less far from 0. We're making it closer to 0. If something is hyperpolarized, then that means we're going to make it even farther away from 0. Okay, we're going to pull that even farther away from 0. We're going to make it, in this case, we're going to make it more negative, typically. Okay. So if resting membrane potential is negative 70 millivolts, if now the membrane potential is negative 40 millivolts, is that a hyperpolarization or a depolarization? It's a depolarization, right? Because negative 40 is closer to 0 than negative 70, right? Okay. If I went to negative 100 millivolts, that would be a hyperpolarization because it would be moving farther away, right, as compared to resting membrane potential.
Can you go the opposite, be overstimulated too?
Yeah, you could overshoot 0, and we'll talk about that. When we do the action potential, we're really going to cap out somewhere around positive 30, positive 40 millivolts, and we're going to work our way back down. So that's going to be that sort of, that's how we're going to know we fired that action potential. If we were to go considerably far above 0. Now, Matt, we don't typically apply the words once we're in that action potential, right? And we're actually talking about action potential. We don't typically talk about like going on to the other side of 0 as a hyperpolarization. We still think about that as like a depolarization. Then there's that action potential event, and then we're going to work back. So typically the hyperpolarization, depolarization, we're talking about before an action potential or after, right? Not while it's going on. Does that make sense? That's actually a really good question. All right, so now we want to think about forces that are going to be moving ions, right? Because what we need to do to make these action potentials happen is we need to move ions. If the inside of the cell is really negative, and to get an action potential, I've already kind of given you this preview, it's going to have to become positive. There are a couple things that we can do to make that happen. One, we could get rid of negative ions on the inside, and that would make us more positive, right? Or we could allow positive ions to come in from the outside, and that would make us more positive, right? Essentially, we have a limited supply of negative ions inside of a cell. It's a small area, right? So I can only let out so many negative ions. In fact, I really can't let out any negative ions. We'll talk about that in a moment. However, do you remember that time I told you that you're basically just a big vat of salt water? I've got an unlimited supply of sodium. a positive ion, right? So what I can do is I can allow sodium to enter the cell. That's going to increase positive, right? And that's going to drive me away from that resting membrane potential toward zero, toward a threshold we're going to call threshold of excitation, toward a certain level where once we get there, we're going to fire that action potential, one way or the other, okay? Now there are a few other sort of forces going on. There's the force of diffusion. We want to think about diffusion, okay? If I open the door to the classroom, guess what happens? Diffusion is going to cause most of you to leave, right? Because the concentration of students is high. Here, when I open the door, you're going to try to bring that to equilibrium, right? So you guys are going to kind of flow out into the world, and then, you know, like student concentration is going to come to some average across campus, right? Think about that. Like at class change times, right? So like at 10:50 on a Wednesday, right? How many of you know 10:50 on Wednesdays exist? Okay, they do. So at 10:50 on Wednesday, like 10:49, assuming your professor is like me and keeps you till the last second, right? You guys are really concentrated in a few places, right? But then we sort of open up, right? And through the process of diffusion, you guys all spread out for 10 minutes, right? And so like diffusion has caused like an equal spread of students across campus. Of course, then by 11, most of you should be back in another classroom, right? So sort of have that. There's also electrostatic pressure. You guys know positives repel, negatives, you know, positive repels positive, positive and negative attract. So if we have sodium, if we have chloride, inside the cell, we're going to have potassium, and we're going to have something called an organic anion. Now that organic anion actually cannot leave the cell, right? It's too big, doesn't have a channel, so it's going to be able to leave. So we're always going to keep a little negative inside the cell. Now that's going to be awesome because we want to get back to that negative membrane voltage, right? That negative resting membrane potential, so we're going to keep that negative in there. But these forces are going to push on each other, right? The sodiums and the potassiums, calciums, all of these electrostatic pressure is going to act. We also have a thing called the sodium-potassium transporter. Sometimes it's called the Sodium-Potassium Pump, also known as the Sodium-Potassium ATPase. Anything that ends in ASE, that's typically an enzyme, that's going to chop things up, right? It's going to break things down. In this case, we're going to break down ATP into ADP, and then that phosphate's going to like fly off. And that's going to be awesome because it's going to release some chemical energy to allow us to operate this pump, right? And essentially what we're doing with this pump is we're moving sodium out of the cell and potassium back into the cell. Okay? And we're going to do this in such a way that we're going to move 3 sodiums out for every 2 potassiums in, which is going to give us a net of sort of 1 negative every time that we operate one of these pumps, right? So that's going to help us get back down to that resting membrane potential at negative 70 millivolts, right? It's going to help us kind of maintain that negative inside of the cell, okay? Hey, there you go. Intracellular, you've got those 2 guys. Extracellular, you have these 2 guys, right? We just talked about that. Not a big deal. Here's an awesome drawing. This is your phospholipid bilayer, that great fluid mosaic, right? You guys have heard of this, hopefully. Okay. This is just showing you outside, inside again, all of your ions, the forces that are on those ions, whether that's diffusion, electrostatic pressure, we talked about that. What's not included here are ion channels. Okay? There are ion channels for those 3 ions, for sodium, for chloride, and for potassium. Okay. Neurotransmitters and drugs that are shaped similarly to neurotransmitters, we'll talk about that later, can open or close these ion channels, which will affect neural activity. Okay? Here's your sodium-potassium pump. Again, we talked about 2 potassiums in for every 3 sodiums out. Again, this is using a ton of energy. This guy's running all the time, okay? Constantly running. So it's constantly using ATP. So you constantly need that, right? Okay. Remember that time that I said your brain only runs on glucose? That's why you constantly want to eat things that are sugary. Okay, this was not a problem prior to the invention of Fun Dip, chocolate, Jolly Ranchers, Coca-Cola. I'm just going to, punch a famous brand in the face there. Back when the sweetest thing you could get your hands on was an apple, it didn't really matter, right? You loved apples, okay, as a species, right? Or any other sort of fruit, right? Because they have sugar, so that's awesome. You can eat your fruits and you're good to go. Now that you can get your hands on other things, right, that have all of these additional sweeteners and sugars in there, then that's one reason they're very palatable, is the word I like to use, right? You ever looked at everything that has sugar in it? How many of you have looked at stuff and you're like, why'd they put sugar in my soup? Right? It's like, Regan, right? I mean, it's like, why do I need sugar in my soup? I don't know, right? I think about it. In my peanut butter. Like, there's sugar in my peanut butter. It's like, I don't need that. Right, Eric? Do you eat peanut butter? Yeah, I eat the peanut butter. The only ingredients is peanuts. Like, literally, that's it. Of course, it takes me half an hour to stir it. And I think the other problem is, so I'm gonna do this. I'm gonna invent my own peanut butter company, but I'm gonna make the jar like this much taller than it needs to be, because invariably I spill the oil, right? That's like on top, and then I'm just left with like paste at the bottom. It's like if it was taller, then I could stir it and actually. I know it's a little extra plastic, but it's okay.
When you want it shorter though, short or flat.
Yeah, but you still need a little stir room at the top, right, to handle. You don't think so?
We'll figure it out.
Draw me up some designs. We'll pitch it to the investors. We'll take it on Shark Tank. I think Mark Cuban's into peanut butter. No. Actually, I saw these guys on there recently. They had something called Nuts and More. It was like a peanut butter protein product. I don't know. Did you see that episode? It was. I happened to be at the gym while it was on the TV. It's like the only time I watch Shark Tank is it's on the TV at the gym. All right. Action potential. If the cell is depolarized enough so it hits that threshold of excitation, we're going to have an action potential. It's going to be this brief electrical impulse, okay? And that's how we're just going to send information down that axon to another cell. Who remembers Johann Mueller? Yeah, and that whole doctrine of specific nerve energies, right? Okay, that electrical signal, that electrical impulse that we're sending is this action potential. They're all the same. Every one of them, give or take, looks something like that. And I mean give or take like my artistic ability, not give or take physics, okay? How we know if it's an apple or a banana is how many of these and in what pattern, right? Okay, so we think about this thing called a rate code. We'll talk about that later. So we've got to hit that threshold of excitation. All right, so here are your ion channels. I know they sort of look like bell peppers, right? This is sort of how they work. They have these different sort of slices, and these chunks can kind of move and shift and open up a pore. That's going to be down the center of that ion channel. That's going to allow ions to flow through. Now, Jasmine, it doesn't matter. You pick out whatever ion channel you want this to be. If you want it to be for sodium, imagine it's for sodium. If you really like chloride, imagine it's for chloride. It doesn't matter. They all operate on the same basic principles. Okay? They're all going to have these moving pieces. They're going to open, and the ions, that specific ion, is going to flow through. Now, the shape and structure of this pore is different from ion channel type to ion channel type, right? So sodium ion channels are structured in a specific way to only allow sodium ions to flow through. Chloride ion channels are only structured in a way to allow chloride ions to flow through. We don't have really a multi-purpose ion channel. We've got one for each kind of ion that can leave or enter the cell, okay? Some of this is going to be based on shape. Some ions are larger than others. Some of it's going to be based on the sequence of charges lining that pore, right? So that's going to allow or inhibit the movement of ions with a positive or a negative charge. Sort of depends on what's going on there. This may be the most important slide from this chapter, and maybe even from your life, right? Because this is the secret to all existence. Okay. Any Douglas Adams fans out there? Yeah. I know he said like 42, but really it was an action potential. He just, you know, it doesn't have the same sort of literary presence, right? Those of you that aren't Douglas Adams fans, you failed. I would suggest becoming one in the next, like, 12 weeks. None of that's going to be. He wrote Hitchhiker's Guide to the Galaxy. That was his most famous work. Anyway, this black line is tracking the membrane potential over time. And when I say over time, this is over like 2 milliseconds. Okay? Action potentials are really fast, really brief. Okay? They're also a word that we call discrete. And I don't mean discrete as in like they're really good at keeping secrets. I mean the other kind of discrete, like they're separate. Okay? And we need these spikes to be separate, right? For the same reason, anybody know Morse code? We talked a little bit about Morse code maybe and stuff, right? So it's all like dots and dashes, right? So let's say you wanted to send a sequence of dots and dashes to indicate that you were stranded, right? SOS, right? You got like dot, 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 dash, 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 dot, dot. It's a pretty simple sequence. What if you decided like, hey, you know what? That's kind of ridiculous. Like I keep like hitting the clicker to send these different signals, right? What if I just like figured out how long like the sequence of dots and dashes was, and I just held the button down the whole time, and then I let up? Doesn't make any sense, does it? Because then you just get, that's not a signal. Like, what does that mean? That can mean anything, right? And so your brain works the same way. Instead of going like a long sort of action potential that would just come up and stay up and then go back down, what we really need is separate independent spikes, right? Because we're going to have to come up, we're going to have to come back down. So, imagine, just briefly, that you are a neuron. You have ion channels. Imagine your dendrites reaching out into space, being tickled by other terminals from various other neurons, right? So Travis, you're really getting into this, I appreciate it. And so you're this neuron and you're going to sit there, right? And you're just hanging out at this negative 70 millivolts. Okay? Now, let's turn on those other cells, right? Those other terminals are going to be releasing neurotransmitters. They're going to be opening and closing ion channels out on your dendrites. Okay? Opening and closing, constantly opening and closing. Now, because they're constantly opening and closing, your membrane potential is going to be fluctuating, right? It's not just going to be flat lined at negative 70, right? You're going to be moving up and down, a little bit at a time, right? Kind of depending on, am I opening sodium channels and allowing positive to come in, or am I opening chloride channels and allowing negative to come in, right? So that's going to make you more negative. The sodium channels are going to make you more positive. Constantly going up and down like that, okay? At some point, it's likely that enough of those neurons that are touching you, which is somewhere between 1 and 100,000, on average you're going to receive about 10,000 inputs from other cells, right? So each neuron usually receives about 10,000 contacts, okay, Abby? Some of them only receive one, and we'll talk about special cases for that. That's really going to be in the visual system. It's sort of a one-to-one relationship. Sometimes they'll receive 100,000 contacts. That's going to be sort of mostly in your cerebellum with Purkinje cells and things like that, okay? But we have these sort of 10,000 contacts. At some point, enough of them are going to get in sort of a sequence where they're going to all be saying, let's let in some sodium channels, okay? That's going to bring your membrane potential up high enough to what we call the threshold of excitation. This is not much. It's really only about a 10 millivolt shift up. Okay? So if your resting membrane potential is around negative 70, as soon as you get up about negative 60 or so, you're going to be at that threshold of excitation. At that point, you're too far. Okay? And you're going to fire an action potential. Okay? not going to be able to stop that. It's sort of, how many of you have ever had a large piece of furniture, or I know you haven't done this, but someone has tried to tip over a vending machine, right? And you get that thing rocking back and forth, right? And you have to build up that momentum, and when you get to a certain point, it's going over, right? Whatever you do, it's going to topple, okay? And you got to get out of the way. It's like that when you get that threshold of excitation. You're building that momentum, and once you get to that point, we've opened enough ion channels, it's going to trigger more ion channels to open, and we're going to have that action potential, whether we want it or not. Okay? So, at that point, the sodium channels are going to open. No surprise, sodium channels enter the cell. Why? Well, the channel has been opened. Now, this is a bidirectional channel. Sodium could go in or out of those channels, right? Where is sodium concentration the highest? Inside or outside? It's outside because it's that big vat of salt water, right? So, electrostatic force, diffusion, it's going to push everything in, right? Imagine that door is an ion channel. If I open that, do you think more students are coming in? No, students are going to go out, right? Because there are more here, and so it's going to push you out. So sodium comes into the cell. Not a big deal. We're still climbing in terms of our membrane potential, right? Once we get up far enough, potassium channels are actually going to open. Okay? We're going to open these potassium channels. Now, where is potassium concentration the highest? It's inside, right? And so when we open the potassium channels, potassium is going to leak out, and we call that a leak. Okay? We're going to leak out a little bit of potassium. Okay. We want to leak out this potassium for a couple of reasons. One, that's going to help us set up a concentration gradient later when we use that sodium-potassium pump, right? It's also going to keep us from overshooting too much, right? So we want to kind of keep this this signal under control, right? We don't want to just continue to increase membrane potential indefinitely. We want to put a stop on that. Leaking out a little bit of potassium can help with that. The other thing that will help is what we call a refractory period. Okay? Sodium channels have a refractory. They have two refractory periods, actually. They have what's called an absolute and a relative refractory period. Okay? So, sodium channel's open. Sodium's coming in. Sodium channel closes, sodium no longer comes in. So we're no longer sort of climbing on that membrane potential, right? Who remembers that potassium leak? Yeah, because potassium is still leaking, the membrane potential then starts to come back down, right? Because we've stopped the influx of positive, but we're allowing that outflow of positive ions, so we're going to start gradually becoming more negative, right? The absolute refractory period is this time period when the sodium channels are closed and they cannot be reopened. No matter what you do, they will not reopen. Okay? For the relative refractory period, which is slightly after that absolute refractory period, you can get the sodium channels to open, but you're going to have to really you have to put a lot of effort into that, right? So you have to have a pretty strong stimulus to make that happen. It's not going to happen on its own. It's going to need a little bit of push, right? So if the stimulation is high enough, you can open those sodium channels again. And the cool thing about that is you can sort of get a bonus action potential out of that, right? And so instead of going all the way up and then all the way back down, we'll talk about that in a minute, you kind of come back down and then right back up, and then right back up, and then right back up. So if you have a lot of action potentials to send in a row, taking advantage of that relative refractory period, right, is going to be pretty good, right? So if the stimulation's high enough, we're going to send a lot of action potentials. We don't have to go all the way back down to resting membrane potential every single time, okay? You just kind of ride that wave at the top. Now notice there's still discrete spikes. There's still separate, measurable, discrete spikes, right? You still measure each one of these independently. They're not running together, okay? Because they have to be separate. They have to stay separate, Emily, so we can get that code, so we know what information is being conveyed. Now, if the stimulation is not high enough, the stimulation ends. We're going to go back down all the way to resting membrane potential. Those sodium or those potassium channels are going to close. Sodium channels are going to reset. Now we are going to do something here called the after hyperpolarization. We're going to slightly overshoot resting membrane potential, but again, that sodium-potassium pump is just going to keep working, and it's going to bring us right back up to that negative 70 millivolts, and we're going to be ready to do it all over again. Assuming that wasn't so exciting we died. That does happen. But it's not going to happen to you, so don't think too hard about it. But you can't overthink it to dying. All right, questions about that? It's not too bad, right? All right.
You hit, I guess, part 6 down there at the end. Is that,
yeah.
Is that where reuptake?
I'm sorry?
Is that where reuptake would begin, or is that only?
For reuptake, we're going to be thinking more about neurotransmitters than we are about ions.
Okay.
But any of the sodium or any of the potassium, in this case, that's not pulled back in through that sodium-potassium pump, some of that might escape and just diffuse away. It's just not going to hang out there. But we are going to be doing that sodium-potassium pump like this whole time, trying to keep it down at that negative 70 millivolts. Don't worry too much about that. That's the rate law. Stronger the stimulus, the more action potentials you get. We sort of said that, right? We've got that covered. Saltatory conduction has nothing to do with salt. It's from the word for to leap, right? So we're kind of leaping, sort of a ballet term, right? So we're not thinking about the salt here. What we're going to do is we're going to jump, and this is important. This is a node of Ranvier, right? Not Ranvier. This guy was, I believe he was a French-Canadian, is the guy who described this, was Ranvier. So these nodes of Ranvier are these gaps between the myelin sheath. This is the opportunity to sort of get a little sort of boost to the action potential, right? This is how your myelin sheath works. So you've got the stimulus. It is going to kind of what we call decremental conduction. But as soon as we get to that gap, we're going to get a boost again, right? We're just going to, and this is how we move really fast from one end of the axon to the other, right? So we send these signals super fast. Not the speed of light, but still pretty fast, right? As Helmholtz proved to us. So, saltatory conduction, that's important. Now, we have sent that electrical signal down the axon. It's made it to the terminal. We do not typically, now this happens sometimes, Jasmine, but not typically, are we going to send an electrical signal from one neuron to another, right? Most neurons are not electrically coupled. Some are. Okay, there are some unique instances where we want these guys to be electrically coupled, okay? But I like to think about this like, how many of you have electricity in your home? Yeah, and how many of you have like multiple switches that operate different lights or appliances? How many of you have different outlets and buttons that you can push to turn things on and off, right? That's pretty awesome. You don't have like one switch that always turns everything on or always turns everything off. That would be bad, right? Because that would mean you would have to like always dry your clothes while you're watching TV because everything has to be on at the same time, right? And that sounds like a bad way to like wire up your house. So Jason, if you're planning on becoming like a home builder at some point, don't wire your home that way. You want separate switches for different appliances. And your brain works in the same way, right? So we want to be able to have like turn some things on, turn some things off. If all of our neurons were electrically connected, when one was excited, that electrical signal would just go everywhere, and then everything would get excited, right? And that seems like a bad way to sort of coordinate things. Although, again, there are a couple places where that is the case, and we'll worry about those later. In general, we do a thing called synaptic transmission, okay? And this is actually how we get a signal from one neuron to another, okay? We're going to take that electrical signal, and we're going to convert it into a chemical signal, okay? And we're going to go across the gap between those two neurons. So we have what's called a presynaptic cell and a postsynaptic cell. The synapse is actually the gap, right? So that's what sometimes people refer to the synapse and they'll include sort of the terminal and the dendrite in there and they'll refer to it all as a synapse. That's fine, but if we talk about the real presynaptic versus postsynaptic, presynaptic is going to be the terminal. Postsynaptic is going to typically be a dendrite, but it can also be an axon. It could be a soma, right? Those terminals can really hit any other part of a neuron, right? Any other postsynaptic neuron. All right, we don't need to talk about that. Here we go. Here's the synapse. Here's what's going on. This is your axon coming down. This is that terminal button at the end. These guys are called synaptic vesicles. Now, what do we have in synaptic vesicles? We have neurotransmitters. What's a neurotransmitter you guys have heard of? Who's heard of dopamine? Yeah, like everybody. How many of you have heard of serotonin? Norepinephrine? Yeah. All of these are neurotransmitters, right? I'm using abbreviations here, but that's fine. There are probably 50 different molecules that have been identified as neurotransmitters, okay, or neuromodulators, like things that are used as signaling molecules in your brain. You're not going to have to know all of them, okay, so don't worry about that. I know you guys are really worried about that, but you don't have to know them all. We're going to talk about the main ones as we go along. In a chapter or so in the future, we'll cover different systems and things. Right now, just know about them sort of in general, like what is a neurotransmitter? Okay. They're stored in these synaptic clefts until an action potential hits these terminals, right? And when an action potential gets down here, that electrical signal starts a cascade of events that's going to release these synaptic vesicles, these little kind of like spheres full of neurotransmitter. And they're going to make their way down to that membrane. They're going to merge with the cell membrane and they're going to dump their contents out into the synapse. Those little molecules in neurotransmitter are going to diffuse across the synapse and they're going to bind to ion channels, to receptors that are connected to ion channels. And those ion channels are going to open and close, allowing sodium or chloride to flow in and out of the cells. And that's how we're going to create another action potential in another cell and then propagate that signal forward. Does that make sense to everybody? It's not bad, right? All right. There are sort of two kinds of receptors we want to think about. Two broad classes of receptors. Okay. One broad class are called ionotropic receptors. These are very direct. They're one-to-one, and their effects are rapid. Okay. They're also sort of spatially constrained. We'll talk about what that means in a minute. Okay. So, we've got an ion channel here. We've sliced this guy in half. Right? So we're kind of looking at half of that bell pepper. We've got our pore in the middle here. Okay. In this case, the pore is closed, so the ion will not fit in. When we have that neurotransmitter, pick your favorite neurotransmitter. We'll go with dopamine. Let's imagine that's dopamine. When dopamine hits that binding site, that's going to start shoving around on those amino acids. And it's going to cause those amino acids to shift, right? And it's going to cause a cascade of shifts, okay? That's going to result in this pore opening and allowing ions to flow through, okay? We call that a conformational change. Okay? It's going to change its conformation, right? The way it's shaped. And it's going to allow those ions to flow through. notice the receptor is tied directly to the ion channel, right? We need one, in this case, one molecule of neurotransmitter to open one channel, okay? It happens right there in that one place, okay? And it's very direct. Right? Now we want to contrast this with the metabotropic receptors. This involves G proteins and the second messenger system. How many of you guys have heard of G-coupled protein receptors or G proteins, second messenger systems, right? This is going to involve a cascade. Okay? And I don't mean the stuff you put in your dishwasher, okay? I sort of mean the thing that, like, you know, you guys think about a cascade, right? You're thinking about like the way water kind of falls, right? Kind of spreads. Or you could think about the way cats breed. That's also a cascade. Okay? How many of you have ever had a cat? Yeah, and that found another cat. And the next thing you know, you had 8 cats. And then you couldn't catch all 8 of those cats and get things tied up properly. So instead of having 8 cats, then the next thing you know, you got 64 cats. Right? Because all of those 8 cats found another cat, and they all had 8 cats. It just keeps spreading, right? That's the way it goes. So you got a cat here, and then each of those cats has 2 cats, even, right? And then, I mean, look at how many cats you have. It's like a lot, right? So this is my Bob Barker message, you know, spay and neuter your pets. Just word to the wise. Cheaper, you'll stop spreading the brain parasites that are making all of you walk out in the middle of the road on 20th Street. I see it happen every day. So there you go. What really is hilarious is when and I've seen this like 3 times, I've seen people get splashed with water there, right? So that's funny. I mean, it's never been me, so it's always been really funny. So there you go. So, what about these metabotropic receptors? Well, you've got your neurotransmitter. Okay, so that's awesome. You still have a receptor, but that receptor is not directly linked to an ion channel. Okay? So, we've got the receptor. It's going to grab that molecule of the neurotransmitter. Then it will send out a signal. Sometimes cyclic AMP, right? That tends to be a guy that's involved. Your G protein is going to activate an enzyme. The enzyme produces the second messenger, and that's going to open the nearby ion channels. Okay? So it's a process. This takes a little bit longer, right? Because we've got these steps involved. Okay? It's not spatially constrained in the sense that these second messengers can travel, right? They're not stuck right there at the receptor. They can go somewhere else. And I know this is like a short diagram, but look, I mean, there's some distance that travels to open those ion channels. It's also not a one-to-one, right? I mean, in this example, each of these molecules is opening 4 or 5 ion channels, right? So instead of just opening in one or closing one, we can open or close several ion channels. Okay, so it's a one-to-many. It's more indirect, right? So it's you've got that sort of time. Of course, it's a bit slower, and it can be we'll call it geographically expansive. Right? So you can spread out a little bit, and you can open ion channels that are separated by some distance. All right, we already talked about this stuff. These are 2 important phrases, EPSP and IPSP. So what is a postsynaptic potential in general? Who remembers the resting membrane potential? Okay. If we hyperpolarize or depolarize that, then we are changing that potential in one direction, and that is postsynaptic, right? If it's after one of these synapses, if it's in response to changes in neurotransmitters and opening and closing ion channels, right? Okay, so that's going to be a postsynaptic potential. If it's excitatory, guess what that means? It moves us closer to an action potential. It may not make an action potential, but it moves us closer and it moves us in that direction. If it's inhibitory, it moves us away from that. It's pretty good, right? IPSP, EPSP, not a big deal. Okay. Yeah, this should actually be on the next slide. My apologies. How do we end a postsynaptic potential? Like, how do we terminate the effects of neurotransmitters? Okay. One way to do this, Matt, you already brought up, is reuptake, right? It's kind of like recycling. Your brain has a number of transporters that can reach out and pull back neurotransmitter that's been released. Okay? And it can recycle that. It'll bring it back in the cell. You can reuse that. You can modify that if you need to. So we've got that reuptake process. The other way is through enzymatic deactivation. Right? And so we'll have enzymes such as acetylcholine esterase that break down acetylcholine. And that, of course, will end the effects of acetylcholine because it no longer exists. So you sort of have two approaches. These two may not make a lot of sense to you, but let me give you some sort of direct applications for things you've heard of. How many of you have heard of SSRIs? That's a selective serotonin reuptake inhibitor, right? Okay. So we are blocking the reuptake process of serotonin. We're not terminating the effect of serotonin. In fact, we're extending the effect of serotonin, right? So we'll talk later, and that's an agonist. So we're going to increase the effect of serotonin, right? How many of you have ever heard of MAOIs? How many of you have ever read a medication label and it said, don't take this if you're on an MAOI? Yeah, right, like everything says that. That's a monoamine oxidase inhibitor. Monoamine oxidase is the enzyme that breaks down monoamines. Monoamines are things like dopamine, for example, is a monoamine. And if we're going to inhibit that enzyme, then we're just going to increase the effect of any monoamines, right? Because we're not going to be breaking them down. They're going to be more likely to stay intact and cause whatever effect it is that they generate in this case, right? So there you go. That's exciting too. Here's just movements. Again, we've got some different ions moving in and out. That's not too big of a deal. Here's that reuptake process, right? These are the transporters for whatever. Let's imagine that's dopamine. So you have the dopamine transporter. Can pull dopamine back into the cell that released it. So you can end the effect of dopamine here at the synapse. All right, what's this idea of neural integration? Who remembers that time I said there were three things? Input, calculation, and output. Neural integration is really the calculation step. Okay? Sort of straightforward. We're going to take any contacts onto that neuron that open sodium channels, and we're going to add or subtract those from the ones that open chloride channels, right? And we're going to see what the end result is. If you get more that say sodium, then you're probably going to be more likely to fire an action potential. If you get more that are chloride, you're going to be less likely to fire an action potential, right? So you're just kind of sort of a summation process where you're going to combine inhibitory and excitatory postsynaptic potentials to see if that results in the creation and firing of an action. We should talk about autoreceptors. These are kind of cool. Your cells have autoreceptors. This is a way that you can monitor release from your cells. So it's really nice to know, did I actually release a lot of dopamine or not? Okay? And if autoreceptors are activated, then we know we've released a lot of that neurotransmitter. If they're not activated, we know we've not released much of it. Okay? And they can have sort of an effect on these internal processes for releasing neurotransmitters. If they're activated, it'll shut off release because we know we've dumped a bunch of dopamine out into the synapse. And we already talked about other types of synapses. Don't worry about that. There are other forms of communication we'll talk about later. Neuromodulators, we'll talk a lot about hormones when we get there. So that's chapter 2. I hope you were all thrilled by that. All right, let's talk a little bit about chapter 3. This is structured. So the last chapter was really focused on sort of that subcellular structure, right? We're going to revisit that in a few, and we'll probably in chapter 4 we'll revisit that. We talk about agonists, antagonists, we'll talk about drug action, pharmacokinetics, things like that. It'll be exciting. But we need to talk about some broader structure for a while. So we'll talk some basic features. We'll cover development just a little bit. Now we'll talk about some developmental things as we go along as well. And we'll talk about central versus peripheral nervous system. Here are some basic terms that most of you should know for directions. I'm not going to really use a lot of these when I'm telling you, like, hey, this is located dorsal or ventral or something else. It's really nice, though, when you read and something is rostral or caudal, which means toward the nose, toward the tail, right? So those are kind of nice things to know. Medial lateral, ipsilateral, and contralateral. That means either same side or opposite side. You'll see these terms a lot when we talk about different brain regions. So just want to throw that out. How many of you have ever cut a brain open? Just like sliced through a brain. Yeah, it's a lot of fun, right? You can actually see a lot of cool stuff, and it's actually sort of a fascinating experience. I don't know, can I ask you the species, Eric? Is that something you'll share?
Sheep.
Sheep, yeah. That's a pretty common one for dissection. Sheep brains are actually shaped a lot like human brains, and sort of the larger structures, when you cut those open, it looks a lot like a human brain would look, kind of on a smaller scale, though. It's actually pretty cool. I've cut a number of sheep brain open myself, kind of see what's on the inside. It's fun. You can cut these in different ways, and that'll reveal sort of different structures. All right, now we have sort of two broad divisions of the nervous system. We've got what's called the central nervous system and the peripheral nervous system. The central nervous system is the brain and the spinal cord. That's pretty easy, right? Brain, spinal cord. It's definitely going to be encased by bone, right? So for the brain, we call that bone the skull. And for the spinal cord, we call that the vertebral column. Okay. The peripheral nervous system is going to be anything that is outside of the brain and the spinal cord. Okay. We're going to put the cranial nerves out there. Occasionally people will debate you on that. Just tell them you're right and they're wrong. We'll put the spinal nerves there, any peripheral ganglia, things like that. Things that are not brain or spinal cord, we're going to call that peripheral nervous system. That's not bad, right? In case you didn't really understand that, here you go. Brain, spinal cord, everything else is going to be peripheral. Here's a human brain. It's a drawing of one at least. Different ways to cut this. We'll talk about cutting like frontal or coronal, horizontal, sagittal. This isn't going to make a huge difference to you, but just to show you that the brain is three-dimensional. Stuff inside of it's three-dimensional, right? And sometimes we're going to need to look at it from different angles so that we can get a full understanding of the structure of the brain and everything that's inside of it. Hey, how many of you love alligators? Oh, right. Really? Well, that's cool. Yeah, so... I'm sorry?
A little one.
A little one, yeah. Well, I'm going to have to tell that part.
I mean, it was still like big, but...
Yeah, well, that's fine. So you have an alligator. Why is it important to know that humans aren't alligators? Well, you know, I mean, there are a lot of obvious reasons. The less obvious reason has to do with what's called the neuraxis, all right, which is the way that your brain and your spinal cord are oriented in your body. So a big difference between like humans and alligators is like 2 feet versus 4 feet, right? And if you see the way that a rat, for example, a cat, a dog, anything that's kind of quadruped, its brain and its spinal cord are plugged in differently, right, than the way that a human brain and then its spinal cord are plugged in. Okay, so when we talk about things being sort of rostral or anterior and caudal or posterior, those are slightly different in humans, but not enough to make you worry. So if you hear about something, you know, in a rat or something, it's probably where we're going to. There are not a lot of like brain studies on alligators. There are a lot of reasons why. One is their brains aren't that well developed. You know, reptiles don't have a lot of the cool things you have in their brains. Alligators are pretty big, right? And they have that long kind of maturational process. So it takes a long time to like get an adult alligator to actually like use. You have to have like a lot of space for them, like compared to a rat, which is smaller, has a better developed brain. All right, we should talk a little bit about the meninges. Who loves meninges? Yeah, right? You can name your kid meninges. Write that down. That was a good name, wasn't it? Honestly, yeah. First kid I'm getting is named the meninges. If you're not planning on having kids, which I think is great, so do that. You can name a cat or a dog meninges. It's always fun. If you have a kid, you can always rename one of them. You don't even have to have another kid, just start calling them meninges. Nobody's going to do that. What about a band? Anybody in a band? Nobody's going to admit to that. But meninges would be a good name for a band, right? Jasmine and the Meninges. I think so, right? I think that's something. So there you go. The meninges are these protective membranes that are around the brain and spinal cord. You've got 3 of these. You've got one that's called the dura mater, which means tough mother. If you've ever, so Eric, you admitted to cutting the brain. I don't know if there was any dura mater left on, sometimes there is, on those sheep brains that you get for like dissections. That's the really tough, you know, like on the outer side, there are probably little bits of it, right? The really tough, kind of strong part. It was really maybe difficult to cut through. That's the dura mater. It's really on the outside. It's flexible, but it is tough. You've got this middle layer called the arachnoid membrane. The reason it's called that is when you sort of pull up, like on the dura mater, it kind of creates these little spider web looking things in between, so it's called the arachnoid membrane. And then the last layer is called the pia. The pia will actually adhere to the surface of the brain. These other two don't, but it adheres to the surface of the brain. So your brain kind of looks like this, right? And you got like some holes here in it. We'll talk about that. We have these convolutions. And those convolutions are called sulci, or sulcus if it's singular, and gyri, or gyrus if it's singular. The gyrus is the bump, sulcus is the valley. The pia will actually go in and out of all of these sort of hills and valleys in your brain, right? So that's kind of exciting. You do have some fluid in your brain called cerebrospinal fluid. It's very similar to blood plasma. You've got some ventricles in your brain. This is going to be, you can deliver nutrients, and it's got some kind of like, you know, filtration process thing, right? To kind of clean up cellular debris and things that's in your brain, drain that out, right? Through your, what we call the venous sinuses, so you can kind of get all of this sort of molecules and things in and out of your brain. Not a big deal. We'll talk a lot about ventricles later when we talk about brain imaging studies, ventricle size, and various neurological disorders and so forth. You actually have 4 ventricles. You're thinking, why is there the lateral and then the third? Because you have 2 of these. So you've got 2 lateral ventricles. And then the next ventricle is going to be the third. There's a tie for first place there amongst your ventricles. Your third ventricle, and that's connected to your 4th ventricle by way of the cerebral aqueduct. Sometimes there's a thing called obstructive hydrocephalus. Hydro meaning water, cephalus referring to your brain. This is when there's a blockage in the flow of cerebrospinal fluid. When this happens, you need a brain valve, right? How many of you have a hot water tank at home? How many of you have ever looked at that and noticed there's like a pressure valve on that, right? If things get too built up, instead of turning your hot water tank into a rocket and shooting it through your roof, that valve will pop off, right? And you'll have some water come out, okay? Which is smart. Believe me, that's why you should always have like a tray or some kind of drain around your hot water tank, right? So it doesn't like blast water all over your floor and ruin everything. So think about that when you're also building your house, Jason. So here are your ventricles. It's a lateral ventricle. Here's your third, and down here's your fourth ventricle. You kind of see it from different perspectives. Not a big deal. Here's a baby. Everybody loves pictures of babies with goofy-looking heads. This particular child, as happens sometimes, there's a blockage. Maybe some of the cerebral aqueduct or some of the other drainage areas have not developed large enough, and so you have to put a valve in the back. So you've got this kind of nice long tube in the brain, a little pop-off valve here. To make this really awesome, you're going to run this tube underneath the skin and dump it out into the abdominal cavity, where that'll just be absorbed into the rest of your body fluid. So it's not too big of, it doesn't sound pleasant. Nobody wants to sign up for a brain valve. I'd like to have one of those. Start thinking too much, you need a little pop-off valve there to release some pressure. It's not exactly what this is. But if you have the buildup of the cerebrospinal fluid, you're not getting drainage, you will have to have a nice little valve inserted. So there you go. Not a big deal. Let's think about development a little bit, right? This is important. We all went through a developmental process, so congratulations on that. It's completed growth phase, whatever, I don't know. It's probably mostly an accomplishment for your parents. I don't think you really were as involved in it as you think. You just kind of existed through the process. All right, Eric, they had to feed you and like provide things for you. Your central nervous system actually starts developing pretty early in embryonic life, and it really just starts out as a hollow tube, right? So it starts out as a hollow tube. And really, if you think about your nervous system even now, it's still a hollow tube, right? Remember those ventricles, that fluid-filled space, that actually feeds into the to your spinal cord, which has a tube going down the middle. So really, your entire sort of central nervous system is just a hollow tube, right? Because you've got this like fluid-filled space in the middle of it. So it doesn't really change. You sort of flesh it out a little bit, but it doesn't really change. It definitely elongates. You get all your pockets and your folds, and it definitely thickens, right? Especially that brain region. Okay? So here you go. I'm not going to ask you a whole lot of questions about this, but just to understand the basic process. Here's where your brain's going to be, up at the top. This is that neural tube that's kind of folding and fusing, like those neural folds. If for some reason, and there are, sometimes there are problems here, right? Where these folds will not fuse properly, or you won't create the neural tube properly, right? So there can be some developmental issues in children. Things that can make this worse are exposure to toxins, right? Things that affect development, right? So I think we all know, like alcohol, for example, is a teratogenic substance that will cause developmental abnormalities, right? If you're as an embryo or a fetus exposed to that. All right, so there's like a developing brain and an embryo. Eventually, that's going to become this guy, right? Which is sort of like an adult brain. Not too big of a deal. That's really all I'm going to ask you about brain development, except for one thing. We didn't talk about synaptic pruning, but we'll talk about that in a moment. Maybe. You do have some subdivisions of the brain. I don't know. I'm one of these people who doesn't think this is terribly helpful. I know there are people who will spend like a long time talking to you about, like, oh, you got to know the divisions of the forebrain, or well, like, who cares that it's the forebrain? It just happens to be in the front, right? That's what makes it the forebrain. Now there's important stuff in the forebrain. I'm not going to say there's not. You got your cortex, basal ganglia, thalamus, hypothalamus, but I mean that's kind of arbitrary deciding what's forebrain and what's midbrain, right? So where are things in the front or the middle, and it's all relative, right? And in fact, most of it's all forebrain because I'll tell you, your thalamus is right here. So like all of this is going to be forebrain. I don't know where that helps me much, right? I don't know. I just don't know where that gets me anywhere. So if you're one of these people who definitely thinks you need to know about this, go ahead. Eloise, have fun with it, but I'm not going to ask you about it. I will ask you about some of these structures, but we're not going to talk about them all today. In detail, we'll talk about them as we get to them, right? As we need to talk about the thalamus, we'll say, hey, time to talk about the thalamus. Hey, this is kind of interesting. This is your cortical development. Just talk a little about that. The idea eventually is to get you 6 plus or minus layers. Now, here's the thing that happens in science. Science is often limited by its methods, right? One reason we've had such major advances in neuroscience in the last few decades is we've had major advances in microscopy. We've had major advances in measuring electrical signals in brains, right? These are not things that were available in the early 1900s, or in some of these things weren't even available in the mid to late 20th century, right? Or they're available now. We can do some cool things. So when you're constrained by methods and you do an experiment, you do a study, and you find some data, and you're like, this is what we have, and so you're going to publish that, and that's awesome, and that's what we have, that's the story. This happens with cortex a lot. So in your cortex, it's that outer kind of crinkly part of your brain. It has 6 layers, except it actually has more. It had 6 layers the first time somebody took a look at it and said, hey, it's got 6 layers. But then we got better techniques, we got better methods, we got better microscopes. We had more specific sort of what we call staining methods, right? Ways that we can delineate features in the brain. And when that happened, we actually picked up a bunch of other layers, particularly like layer 4. Layer 4 probably has 5 layers in it itself, you know, and it kind of depends on how you want to think about this. The problem is like this particular 6-layer setup had existed for like 100 years, and so we weren't going to change it. So instead of adding like, now you have layer 7 or 8 or 9, you've got 6 layers, but you've also got like 4 alpha and 4 beta and 4A, B, C, 1 alpha, and you have like sublayers and sublayers of sublayers, right? So that can be a little bit of a confusing system, but we just decided to stick with the 6 layers, just cram stuff in. I don't know. What are you going to do, right? It's the best we had. Oh, this is kind of cool. We should talk about this. With cortex, we talked about there are things called progenitor cells. These are like stem cells that give rise to the central nervous system. Right? There's a lot of people who are doing stem cell work, trying to find ways to make neurons, make specific kinds of neurons. Right? So that's cool. That might get somewhere. Right? There's a lot of work going on in that field, which is kind of cool. There's sort of a competing field, which is people who are making like brain-machine interfaces, which is kind of cool, right? So you can just use your brain to control robotic arms. I don't know if that excites anybody. I watched a monkey once move a robotic arm, just thinking about moving a robotic arm. So that was kind of cool. I don't know. You can do this. How many of you have a cat? You can probably train your cat to like move things just, you know, like, you don't have to cut its head open. And you have to put in some electrodes to measure electrical signals. But you can seal it up with like dental cement. That's typically how they do this. And the hair will grow back. Just like comb it over.
So was that the monkey and the rolling arm, was that measured from the brain or was it measured from where?
Yeah, it was brain actually. So that's actually, so it's not like the monkey's like moving its arm and we're like mimicking that, right? So the monkey, how they did this, was actually a really good question. So you had a monkey, I'm not going to draw a picture of a monkey, I'm going to try. Got to put a tail. It's a happy monkey. And so then it's just like sitting in this chair, right? And it's actually, so robotic arms, for whatever reason, really freak out monkeys. You know, like if you were a monkey and you were sitting there and all of a sudden this like robotic arm started moving, it really might freak you out, right? So what the monkeys were trained to do is they had a computer screen. And on that computer screen there was like a target and there was a dot. Okay? And those monkeys were trained to sort of use their motor cortex, like their premotor area, we'll talk about this later, like their motor planning area, to plan and think about the movements they would make with their arms to reach out and move that dot to the target. Now in another separate room, was the robotic arm. This is the robotic arm. It's got like these robotic fingers. And then that robotic arm would move and reach out and like grab a ball. So, but it was still coming from motor cortex. It wasn't from like measuring their arms, and it wasn't even measuring eye movements. Because you would think, well, just look at the eye movements. But it was measuring like the premotor, the motor planning area for these monkeys. Now it took a little bit of training, but it was still cool to be able to read, what the monkeys intended to do, and then translate that into actual movements. Yeah, it was actually kind of exciting. All right. Development of complex brains. Don't worry about this too much. Oh, yeah, we should talk about this, though. Overproduction and refinement. This is what we like to call synaptic pruning. You have fewer synapses now than when you were like a child. And that's not because you're like losing things that you need. It's because you're losing things you don't need, right? How many of you have ever been on a camping trip? Yeah. And how many of you took more than you ever needed, right? And that first time you took too much, and you're like, why in the world did I bring shower shoes? There's not even a shower here, so I don't need this, right? Why did I do that? You tried to bring a television set. I don't know, I'm just thinking of ridiculous things. So that first time you take a bunch of things, like, I didn't need that. So what do you do the next time? You take fewer things, right? You take fewer things, and finally you get to the point where you're like, I got it. This is exactly what I need to go on an efficient camping trip, right? These are the items I need. Nothing's going to happen. I'm going to be prepared for, most of the eventualities. Anything beyond this is just going to kill me, so there's no point like worrying about it, right? That's kind of what happens when you're a child. You've got all these extra synapses, and you don't use them. Okay, and if you don't use them for a while, then you just, there's no point keeping them. It's very metabolically demanding, right? Jared, to hang on to this extra stuff. To like carry all of this extra stuff, like out on the trail to camp, when in fact you're never going to need it, right? So you just leave it at home. So it's much more efficient, and you still get the same benefit, right? And your brain's going to do the same thing.
In the last class, we talked about that, and he said that if you didn't have the pruning, then it could lead to mental retardation.
Yeah, it can actually, because you're going to have too many connections, and the connections are not going to be related to anything you're going to need, right, or anything you're going to be doing. So you're going to have all these like sort of aberrant connections, and it's not a great situation. But it's a normal process. You're going to have them, you're going to get rid of them, that's fine. So you really want to go through this process.
That's the same where it's like, you're talking about, we have those inhibitors that keep everything from firing at once, and getting everything excited. Is that, in that case, is that where everything kind of fires too much and we can't process?
Yeah, so this could definitely happen if you have too many connections. They may go to the wrong place, right? So if you're like, I'm going to move my left arm, and then you've got all these extra synapses that are maybe accidentally connecting your right arm, right? And then all of a sudden you're just, why did my right arm move? That's why you've seen babies, right? And you touch them and there's like some random assortment of movements, right? Because they've got all these like extra connections going everywhere, and they don't make any sense. You got to get rid of those. Right? Eric, you've seen a baby, right? That's what they do, right? You poke them in one place and the other side moves. It's like, well, that doesn't make any sense. That's not helpful, right? So you have to prune that out. So that's pretty good. What are some things that do affect development? Hey, here's a shocker. Genetics, right? Nobody ever thought about that. or personal experience, right? Both of those can affect your developmental patterns. Neurogenesis is kind of cool. You still do some of this even as adults. Mostly it's going to be confined to your hippocampus. And your olfactory epithelium, we'll talk about those later. But there is a lot of neurogenesis as a child, right? Remember that neoteny concept where you're born with that sort of underdeveloped brain? We've got to rapidly expand that, and then we're going to kind of narrow down what we need. So here you go. Here is a kind of a picture of that synaptic pruning. You've got your birth, then your, like at 6 years, look at how many connections you have, and by the time you're 14, you've cleaned up a lot of that, right? So you don't need it anymore. You don't need to move your right arm and your left arm at the same time, so they don't need to be connected, right? So we can get rid of some of those connections. Be very efficient, and that's what you want. You want to be efficient because otherwise it takes too long to respond to something. And who remembers that rock that was flying at their face? Yeah, I've not forgotten about it. And if it takes too long for you to get the right sequence of actions to get out of the way of that rock, then you're going to die because the rock's going to hit you in the head. So there you go, right? So you should eliminate as much of your brain as possible. Some people go too far. No. It's a normal process, you're going to be fine. Synaptic pruning. It's awesome. Don't worry about that. All right, here we go. We're going to talk about these things briefly. So you've heard the words, right? We've talked about cortex already. Cerebral hemispheres, that's kind of interesting. Sulcus gyrus, we talked about that. Spinal cord. Hey, how many of you have ever seen a spinal cord? Yeah, they're pretty cool, right? They're actually super small, right? It's like the size of your pinky. You think like, that's it? That's all I got right there for a spinal cord. They also don't go all the way down, right? They stop about 2/3 of the way down your back, right? And from then on, you have a thing called the cauda equina. That means horse's tail. So all the rest of the nerves and things come out. And at the very bottom of that, there's a thing called the filum terminale, right? Terminal fiber that's going to hold your spinal cord down. You don't want that thing to break, because then your spinal cord kind of rolls up. True story, it can actually, if that breaks, it's going to like pull your spinal cord up, it'll kind of retract, and you'll have a thing called shearing, because all of your nerves are coming out off of your spinal cord between those vertebrae. Slice them off. I see a lot of people, Jason, after I tell them that, they walk out of class like this. Just take it easy. I wouldn't think you have to like change your behavior or anything. To break, that's pretty difficult, and you're going to have some other problems related to that if that happens to break. So no worries. Keep doing whatever stupid stuff it is you do. You'll be fine. Spinal cord. Hey, your cortex, you've got things like your visual cortex, auditory cortex, somatosensory, motor cortex. We'll talk about all those things individually. Not a big deal. Hey, here's like a real shocker. Your visual cortex, guess what? Abby, it's involved in processing visual signals. I know you never knew that based on the name of it, but there you go. This is your forebrain, and this is why I said, who cares? It's all the forebrain, right? You've got the forebrain, and then this little part back here that keeps you alive, and this little part right here that keeps you from falling down. And that's really your brain, right? So talking about forebrain, midbrain, hindbrain, I don't see where it gets me anywhere. Oh, we should talk about white matter and gray matter. How many of you have heard of white matter? Okay. It's white matter if when you slice open the brain, it actually looks a little bit whitish. And that's because of the myelin, right? Myelin is a lipid-based, it's sort of a fatty substance, right? Nobody likes to be called a fat brain, but you know, it would be the worst thing you could be called, right? And if you have like a lot of fat in your brain, then that means you're sending your signals quickly, right? I don't know. Doesn't exactly work that way, but something to keep in mind. So white matter is going to be the axons. Gray matter, on the other hand, is going to be like cell bodies. Okay. Here is a gyrus. That's a bump. Here's a sulcus, that's a little valley. If it's a huge valley, it's called a fissure. It's like a bigger hole. Okay. You've only got a couple fissures, you got a lot of sulci. All right, how many of you know about the 4 lobes of the cortex? Yeah. Frontal, occipital, temporal, parietal. Not a big deal. I'm not going to ask you about it. Here you go. Here are different divisions of your cortex. Again, we talked about like visual cortex, somatosensory, auditory. Here's your sort of motor and somatosensory cortex. Here's your frontal cortex. This is also known as the non-reptile cortex. Actually, all of this is sort of mostly non-reptile. Reptiles really are only sort of like from the basal ganglia down. That's really all they have. They don't really have a cortex. Rats, on the other hand, have most of this. They don't get much of this guy in the front. This is the stuff that lets you use an iPhone. Okay? Rats don't really use iPhones. You could probably train one to use an iPhone. I'm thinking about that. I saw a rat play basketball one time, which was pretty awesome. It was like 2 rats. They played one-on-one against each other. They used the ball out of a roll-on deodorant. And there was no dribbling, so it was very old school. No 3-point line. So they would kind of pick up the ball and like go over to this goal, like stand up, and it was all slam dunks. And then they would get a little food pellet for a reward. Actually kind of cool, Eric. I don't know if you have 2 rats, you can train them to do that.
What's the core regulation?
It was scaled down to rat size. So yeah. You should have seen the jump ball. It was really... It's like an extra kind of player control foul, illegal use of the tail. You have to watch out for that. Take a cheese break. I don't remember there being a lot of rules. They just kind of took turns grabbing the ball and like going back and getting there. No defense either. So definitely modeled after the NBA. No defense. It's all offense. We talked about this stuff. Oh, hey, who knows about the corpus callosum? That's kind of a cool thing. Guess what? Most of you have 2 hemispheres. I can't see in your brains or in your head, so I'm going to assume you all have 2 hemispheres. Most of you probably have a corpus callosum. Now, a number of you may not, right? So there's like some evidence, maybe between 1 in 600 or 1 in 1,000 people are born without a corpus callosum. So it's quite possible somebody in this class doesn't have a corpus callosum. You may not know that unless you've had an MRI. To determine that. Corpus callosum is this big fiber bundle that connects the left hemisphere and the right hemisphere, which is pretty awesome. Like if you've ever wanted both halves of your body to know what you're doing at the same time, it's really important for both halves of your brain to talk to each other, right? Otherwise, I see people that try to run in 2 directions at once, and it doesn't really work. The cool thing about the corpus callosum is sometimes you have to have this cut. And sometimes you have to have it cut because you have intractable epilepsy, right? And so this is a really extreme sort of approach to this. But they will sometimes cut your corpus callosum, separate your left and your right hemispheres. And this kind of technique was really pioneered by a guy, Michael Gazzaniga, and him and Roger Sperry. Sperry won the Nobel Prize, Gazzaniga didn't. There was a little battle about that, but that didn't really result in anything. Gazzaniga later was like, hey, I'm an awesome neuroscience guy too, so who cares? I've got all kinds of money and I write books, so I'm not going to complain. They've been pretty good about it since then. But there's some funky stuff you can do with people. If you cut their corpus callosum, you can do cool things like put a partition so they can only see like on one side and what's on the other side, and they won't be able to like communicate from one side to the other, which is actually kind of cool. So you can do some stuff like have them smell things, because odor doesn't cross left to right, it goes on the same side, so it doesn't use the corpus callosum, like vision does though. So if they smell something in one nostril and you're like showing them a picture, they'll be like, I don't know what I'm smelling, I don't see anything, but then you ask them to reach out and grab it and they can do it. The other cool thing you can do with them is, and this will tell you like how people will just make up stories, confabulate. Right, you guys know about confabulation? This is an awesome concept. How many of you like something, but you have no logical reason to support that? But, right, Jared, but then you don't do it. but other people do. But then they'll make up a reason, like a logical reason why they should like it, right? And they'll like really argue with you on this, and you're like, whatever, right? I mean, that doesn't make any sense. It's called confabulation, and you do it all the time. Everybody does. But you do it so much if you've had a colosectomy, you've had your corpus callosum cut. It's cool. To one hemisphere, show them a sign that says "Eat a banana." Okay, and then on the other side, they left eye, right eye. They can't see what there's no overlap, right? So you show them this sign that says "Eat a banana." Okay, so they'll reach out and they'll grab a banana and they'll start eating it. Then you ask them, why are you eating that banana? And they'll say, well, I must have been hungry. They won't tell you that they actually saw this sign because they won't have been able to send that language interpretation where they read the sign from one hemisphere to the other to comprehend so that they can tell you what they're doing. So they'll make up a story about why they were eating a banana. People eat a lot of bananas because they see a sign that says eat a banana. And in fact, they'll tell you that they're hungry. It's just, it's really fascinating. So not only are you controlled by your cat's feces, you're eating bananas and you have no idea why. You guys are going to walk out of here with some existential crisis. It's like, I don't even know if I exist. I'm going to try to walk through this wall. I'd love to see you try it. Because I know you exist, at least as much as I care to know. Hey, these are axons. That's pretty cool, right? Somebody's colored them in with a colored pencil. And you can see these axons coming out. That's the corpus callosum, connecting all of these. And they're color-coded based on frontal cortex, vision, auditory, coming down here in purple, right? So it's different regions of the of cerebral cortex. That's not so important. The limbic system. This guy's important. So, the limbic system involves a lot of places. Things called the cingulate gyrus, limbic cortex. It also involves the hippocampus. This guy's going to be involved in learning and memory. I don't know if you guys have ever tried to do one of those two things. Tried to learn something and or remember it. It's going to be really helpful if you'll do that in this class. I promise. And the rest of your life. This is good life lesson. It also involves the amygdala. The amygdala is going to be fear-related responses. We'll actually spend a lot of time talking about the amygdala in this class. There's 2 or 3 chapters about that, which is going to be pretty exciting. And here's where all your limbic system stuff's located, kind of in the middle. The basal ganglia. These guys are important too. The basal ganglia are really involved in a lot of information processing for motor movements. Individuals who have Parkinson's disease, Huntington's disease, Tourette's, OCD, autism, probably they have a disordered basal ganglia. Some of those guaranteed they do. We definitely know what happens in Parkinson's and Huntington's disease, but it seems as though individuals with OCD or autism or Tourette's also have. And if you think about, there are motor components to those particular disorders, right? With autism, you have sort of this stereotyped behavior. Individuals who have autism spectrum disorder, they'll sort of repeat the same behaviors over and over. I had a neighbor who was autistic, and every time he talked to you, he just rocked back and forth the whole time he talked to you, right? So he had this kind of stereotyped behavior. The whole time he was asking you if he could get the electronics out of your trash can so he could make solar-powered Christmas lights. So he would walk around our neighborhood and take all of our stuff that we threw away that was electronics and make Christmas lights. His parents let him take his whole garage. It was always computers. I mean, I don't know what he did all day. He also had this thing where he would, he had this brief for 5 minutes nightcrawler business where he would sell night crawlers to people. But he took this, those things that cook hot dogs from the inside out you see at gas stations. He took one of those and converted it into a device you could stick in the ground and sort of shock the worms so they would come up to the surface, which was kind of fascinating. It's like watching them. So it's kind of fun. Tourette's, again, that's most people think about Tourette's as screaming obscenities uncontrollably, but it's really usually tics, right? It's motor tics, facial tics, things like that. Again, it's that motor issue. And OCD is kind of the same thing, right? If you think about people who have obsessions, but their compulsions are going to be motor, right? So they're going to wash their hands, they're going to check the doorknob, continuously make sure they turned off the stove, go across and hit the stove buttons, right, to make sure everything's turned off. So those are motor behaviors. And so there's some evidence that the basal ganglia is involved with that. We also have the thalamus and the hypothalamus. Those guys are kind of cool. Thalamus is going to handle a lot of sensory information. We'll talk about that. Hypothalamus may be one of the most important regions in your brain. A lot of people don't think about it. It controls circadian rhythms. We're going to be talking about, well, yeah, the 4 F's here: fighting, fleeing, feeding, and reproducing. Those are pretty important behaviors. Hypothalamus is going to be involved in a lot of motor behavior, or a lot of hormones, right? So it creates hormones. That causes other hormones to be released. So we'll talk about that. It's going to be exciting. We'll talk a lot about that when we talk about the amygdala hormones. We'll talk about development a little bit. Prenatal androgenization. It's going to be exciting. Somebody just got the 4th F. Yeah, it just took a moment. That's okay. That's not my joke. It's probably, I think it's midbrain, don't worry about this. We're not going to talk much about superior colliculus or inferior colliculus. Don't worry about it. Out of the tegmentum, really the substantia nigra is what we'll talk about the most. This guy affects to the basal ganglia. It's the dopaminergic neurons in this projection that die if you have Parkinson's disease. Hindbrain. Hey, this has got the cerebellum, the pons, and the medulla oblongata. These are responsible so you don't die. Right? Your brain knows you're an idiot. It really does. So it's going to try to take away all of the important things from you, like keeping your heart beating, breathing, right? Monitoring your blood pressure, all of those things. Like your pons is going to be, and your medulla are going to be responsible for controlling some of those things. Cerebellum is going to be a lot of coordinating movements, especially making sure that you have completed a sequence of movements you intended to complete. Right? So if you meant to move forward, did you actually move forward? And your cerebellum is going to get that feedback to know. Right? This is one of the first regions to be affected by alcohol, which is one of the reasons why individuals who ingest alcohol, right, in some quantity, sort of, despite the fact they may think they have retained or enhanced their athletic prowess, have actually lost some control of their bodies. Right? So keep that in mind. There's your spinal cord. We will not talk a lot about spinal cord in this class. We'll talk a little bit about it, but not a whole lot. You don't really need to know much about the organization of the spinal cord. Cranial nerves. Really what you need to know about cranial nerves is we'll talk about them if we need to. They do have sensory and motor functions. Some only sensory. Not a big deal. We'll talk about them as we need to. There's the vagus nerve. That really is a big one. That's why it's vagus. It's from the same root word as vagrant, which kind of means to wander the streets, right? And it actually regulates functions of a lot of organs in your abdominal cavity, right? So, things to think about there. What about your autonomic nervous system? We should talk about that briefly, right? That's an important one. This guy is the one who gets keyed up if you if you're in a situation where you need to expend energy, or you've been frightened, there's some arousal sort of thing. So your autonomic nervous system, the sympathetic division, is going to become activated. The parasympathetic nervous division of the autonomic nervous system is going to kind of bring you back down. So this is sort of rest and digest kind of stuff, whereas the sympathetic is more other situations, right? Stressful situations. There are your cranial nerves, spinal cord, not a big deal. Questions about Great. And I know that was pretty fast. Yeah, I think we can stop there for the night, actually.